Good morning, everybody, and thanks to OAS for allowing us to share with you today, the first time to the industry, the results of our first registration study of Azura MD001, a novel keratolytic agent. So at Azura, as I said, we're pioneering a new class of ophthalmic treatments, ophthalmic keratolytics, that really focus on addressing what's going on in the glandular structures as well as the lid margin disease. Our lead program targets meibomian gland dysfunction, and as I said, I'll share with you the results of our first registration study. Meibomian gland dysfunction is really at the top of a lot of downstream effects. It impacts tear film stability, which then leads to reduced vision quality and vision functioning. It's the number one cause of dry eye disease. It also contributes to people not wanting to wear their contact lenses as long as they can and also ultimately drop out. These glandular changes over time can lead to glandular blockage, atrophy, and also overall bacterial growth, and lead to an inflammatory state in terms of blepharitis and ultimately ocular surface inflammation. Meibomian gland dysfunction affects a lot of people, and recently when we shared with our scientific advisory board the data from our phase 2B study, everybody said about 70% of their patients that walk through their door has some form of meibomian gland dysfunction. So it's a very large market opportunity. Azura MD001 is not an anti-inflammatory, it's a keratolytic agent. Its mechanism of action is well known. It's a keratostatic, so it actually slows down the cell replication cycle, particularly keratinocytes, and therefore reducing the production of future keratin proteins in the meibum. It's a keratolytic. It breaks disulfide bonds. It's a fairly potent disulfide bond disruptor. But one of the interesting opportunities we have with this compound is a common side effect in its use as a medicated shampoo. It actually increases lipid production from the sebaceous glands. And we believe it does that by actually increasing the maturation rate of the acini cells. So in terms of a disease that has blocked glands and poor quality secretions, we believe that Azura MD001 has a really unique mechanism of action. So our Celestial study, our first registration study, was a 3-arm study, a 3-month endpoint, 245 patients across Australia, New Zealand and Canada. It was a double-masked, multi-centre, vehicle-controlled study. One thing to note, we had 62% of patients at baseline had level 1 or level 2 corneal staining. The drug is an ointment. It's applied to the lower eyelids at bedtime 2 times per week. So just make a note of that. So in terms of our primary endpoint was the number of glands that were open. We used the meibomian glands yielding liquid secretion, and as you can see, we clearly hit a statistical significance at day 90. But one thing to note was a statistical significance was achieved as early as day 14, and that's with only 4 applications of drug. In terms of the ocular surface symptom measure, we used the Ocular Surface Disease Index, and as you can see here, a statistical significant difference relative to vehicle was achieved. The minimally important clinical difference in this group of people was 4.5, and as you can see, we achieved that. In a more severe patient population, it's around 7.5, and we're getting that sort of range by month 3. But that's, from a regulatory perspective, we're able to achieve the regulatory hurdle. But what I think is really important is the clinical relevance. So in terms of the pre-specified responder rate analysis, we looked at the number of glands that were opening. So the literature supports greater than 5 glands from baseline. And we saw at 3 months, close on 46% of patients achieving that level after 3 months of treatment. When we look at the quality of the meibum, now the International Tear Film and Ocular Surface Society define meibomian gland dysfunction as anybody who has an MGS score of less than 12. And so we achieved close to 70% of patients achieving essentially no meibomian gland dysfunction. And what's I think more important, as early as day 14 with only 4 applications, 20% of patients had actually achieved greater than 12 on their meibomian gland score. So a very rapid, significant clinical response is achieved. But at the end of the day, we just want to make patients feel better and remove their symptoms. We chose ocular surface disease index as the primary endpoint because it had validated cutoffs for symptom level. And any score under 13 is considered a patient who is asymptomatic. And so after 3 months of treatment, we saw close to 50% of patients who actually were asymptomatic at the end of this 3-month treatment. So in terms of adverse events, it's a keratolytic. When you put it on the skin to treat comedonal acne and so forth, you get a bit of burning and stinging, and we saw that. In terms of total treatment of adverse events, 93% of patients or adverse events were mild to moderate, and we only had 2 patients in the 0.5% and 1 patient in the 1% withdraw due to adverse events. So overall, the adverse events were mild and transient in nature. So in summary, we hit our co-primary endpoint for the first ever investigational drug to treat meibomian gland dysfunction in a single study. I think we've clearly demonstrated that a mechanism of action that actually opens the glands and improves the quality of the meibum clearly is demonstrated in terms of the percentage of patients that were able to achieve these cutoffs. And as I said, the clinical relevance of this result, with close to 50% of patients on OSDI becoming asymptomatic. We also looked at tear film stability, which is the natural thing that you would expect to happen when you improve meibum. We improved tear breakup time, as well as the percentage of patients who actually achieve greater than 10 seconds of tear film breakup time. But also, what's more importantly, the consistency of the data across multiple patient-reported outcomes. We had success in OSDI, speed, average VAS, eye dryness, ocular itch, and also ocular discomfort. So with that, we have a clear path to approval. We intend to start our phase 3, our last registration study in the middle of next year. Thank you.